Howdy, and welcome to the Flies facility at Texas A&M University. We are so excited for you to come along with us and learn a little more about what we do. The Flies facility is led by Dr. Jeffrey Tomberlin, and among other things, we study the science of decomposition, which has applications in waste recycling, insect ecology, and forensic science. While this video is family-friendly, we wanted to give you a heads up that some images might seem a little yucky, but we hope you enjoy your time with us learning about the research taking place at the Flies facility.
We are creating waste every day. According to a recent survey, 20% of what goes into the landfill are food waste. We are generating 35 million tons of organic waste each year in USA. Food just being thrown away because people buy more than they need, or the food expired in the supermarkets. At the same time, there are so many people under starvation, and it is projected that by 2050, we will need 50% more food, and there will be a global food shortage. Today, we have a novel biosolution for this issue, the black soldier fly.
Black soldier flies are large black flies that are found in tropical and subtropical climates. They have iridescent blue wings and look similar to wasps, but they can't bite or sting. Black soldier flies are not only harmless, but are also helpful friends of our local ecosystems. In this room, there's about 100,000 flies. Now, if we estimate that half of those flies are female, and each female can produce an average of 500 eggs in her lifetime, when all those eggs hatch and become larvae, they're able to convert 25 tons of waste. A single larva can consume 6 to 10 times of waste in comparison to their body mass within just 10 days. If you watch this video of how fast they're consuming the fish, that's pretty amazing. With just 1 gram of eggs, you can digest up to 10 kilos of organic waste. The black soldier fly is considered to be a detritivore, which means that it can eat many types of decaying matter. And this will include things like produce, grains, meat items, and even manure.
Black soldier flies are one of the edible insects suggested for human consumption by the Food and Agriculture Organization.
They do have a bit of a nutty flavor or taste similar to an unseasoned Cheeto, but currently most of the black soldier fly larvae produced are used for various types of animal feed. So as a general rule of thumb, black soldier fly larvae contain up to about 40% crude protein, about 35% lipids, and they're also very calcium rich, which makes them a very nutrient-dense feed option for many types of animals. This will include poultry, swine, fish, reptilian pets, and even more traditional house pets such as cats and dogs.
However, we already know animal manure is a horrible issue with other livestock rearing facilities.
So not only can black soldier fly larvae be used to eat the manure from other livestock facilities, but their frass, or their poop, which is left over from the larval feeding, turns out to be a really good organic fertilizer. So that frass can suppress plant pathogens, repel pests, and boost production in systems ranging from small home gardens to commercial crops.
So it is a circular economic system. And this is why, along with the increasing research papers published, more and more black soldier fly companies are popping up all over the world. Let's do something good for our planet.
When an animal dies, there are a number of organisms that use it for food. This helps recycle those nutrients back into the ecosystem. These consumers take care of those remains so we as humans don't have to. Without them, the world would be covered with dead things. Understanding the relationships between the different organisms that use these remains as food provides us with useful information about how the decomposition process works.
You might have seen some of these organisms feasting along the side of the road. Scavengers are a big part of the decomposition process. In addition to vertebrate scavengers, like these red kites here, insects also help consume these dead things, including flies, beetles, ants, and even cockroaches.
All of these organisms find remains to be very nutritious, so they will compete to have the resource all to themselves. There are even some competitors that we can't see with our eyes. Bacteria and fungi are super small organisms that also need to eat, and animal remains are the perfect food for them since bacteria are already inside helping the animal digest their own food. When they are not competing with bigger organisms, they can completely take over. We work to understand the dynamics between all of the organisms competing for the resources such as wild boars, brown rats, flies, and beetles. Each of these animals can play a different role in the decomposition process and can affect how quickly or slowly these remains disappear.
In our lab, we focus on blowflies, which are usually a shiny blue, green, or bronze. You can usually see them flying around your food if you leave it sitting out, so you've probably seen them before. You may have also seen their offspring on trash if it's been left out too long. These are the creamy white worm-like creatures that you see here, often called maggots or larvae. Females can find an animal soon after it dies and will lay their eggs on that resource, so when they hatch, the larvae can consume all of the flesh until there's only bones left.
This can be used in a number of ways, especially solving crime.
Forensic entomologists use this knowledge of the decomposition process and understanding of insect biology to assist in forensic investigations. Blowflies are the primary insects we use because we can figure out how old their larvae are and use that information to create a timeline to work backwards to determine when they were laid.
This is important because oftentimes we are asked to calculate how long insect evidence has been present on remains, which can help investigators determine a time of death. Our calculations can also be used in cases that don't involve death, including cases of abuse, neglect, veterinary medicine, and even insects in your food.
So even though we know you might think decomposition is gross, now you know all the ways it can help us. From ending world hunger to solving crime, the Flies facility continues to push decomposition science forward to make the world a better place.
everybody, my name's Melanie Florkowski, and I'm a graduate student in Jessica Yerzinski's lab at Texas A&M University. Our lab studies animal behavior, and with us we have a couple of our peacocks that we're going to show you today. So we have approximately 35 peafowl in captivity. These are some males and females feeding on some corn on the ground. So all of these individuals we have in captivity are going to be adults that were captured from the wild. Most of them come from either populations in California and Florida. And over the years, they've been used in many different experiments and have contributed greatly to our understanding of their biology. Because these birds were all captured as adults, we don't know exactly how old they are. However, in the wild, peafowl can live to be up to 20 years old, and it's likely that a lot of these birds are actually in their mid-teens. So this is a male peafowl, what we call a peacock, and these guys are very colorful and they have these long, elaborate tails. And they use these tails to attract the females. And the reason that they grow these tails for the females is because it indicates that they're very strong and healthy, so the females will know which males are the best mates to choose. The males only display to females during certain parts of the year, so here is a few images of their display and the females evaluating their display. So here we have a female peafowl, so we call these girls peahens, and she's a lot less colorful than the males. As you can see, she doesn't have that elaborate tail that the males have, although she does have some coloration up on her head and neck. It's green. So what we studied is how these females perceived the males' tails and what they looked for when they were searching for a mate. And so we were really interested in how they perceived the world through their eyes. One of the ways that we studied how females choose their mates is actually by putting cameras on them. So we made this special little helmet that goes on the head of the peacock, and it actually has a camera attached to it. And we can see where the female is looking while she's evaluating a male display. So we found that females tend to choose the males that have the most eye spots. So if we look at one of these male feathers, we have this spot at the top, and it's called an eye spot because it kind of looks like an eye. And we found that females choose males that have a lot of these eye spots, and they deem those males to be the strongest and healthiest males in order to raise their chicks. So here is a female wearing that helmet that we were just showing. And we also have some video footage of what it looks like when the birds are actually wearing these cameras. So we have one camera that is pointing at the eye of the female and another pointing out in front of her, and it's showing what she is seeing through her eyes. In this case, she's evaluating a male display in front of her. This technology can be used in other bird species as well. Here we have a great-tailed grackle wearing a modified version of this eye camera. We use it to measure how they use their gaze while they're flying.
Hi, my name is Ming Ray. I'm a PhD student in the Psych and Brain Sciences departments, and now I study birds. This is an example experiment that we're running with the birds, where we're teaching them how to use a computer monitor and looking at their performance in a learning and attention task.
Once the birds learn how to use these screens, we'll measure their brain activity as they perform their tasks. So by studying the vision and learning ability of peacocks is just a couple of the ways that we study evolution and behavior here at the Yerzinski lab.
I'm Bridget Benedict. I'm a PhD candidate in the Department of Ecology and Conservation Biology. And today I'm going to tell you a little bit about Dr. Barbosa's lab, the lab that I work in. Specifically, I'm going to talk about some field methods we use in the lab. Most recently, we've been working on ungulates, flies, and bats, so I'll focus on those. So what is an ungulate? An ungulate is just a mammal with hooves. It's like a rhinoceros, a zebra, a horse, a bison. Those are all ungulates. Here's an ungulate. This is a moose, and this is me, and I'm sweep netting flies off of the moose. So I'm just using this big net to try and collect all of the insects that are flying around the moose. It's another picture of me waiting for this moose to hold still so I can come get some flies off of this one as well. Sometimes I wait for them to lay down. That's usually the easier way to get flies off of a moose. So I wait till they take a nap, and then I can use this big insect net and get the flies off of them. I should also note that you never want to do anything like this with a wild moose. These moose are hand raised, and so they're used to us. And here are some moose calves that I'm using an insect aspirator. So what that means is just basically an insect vacuum. Use this machine to vacuum insects off of the moose so that I can have the insects. I also look at the insects in the habitats that the moose live in. So basically where the moose lives. I want to know what insects are flying around where a moose might be hanging out. So on the left is a sticky trap, that thing that kind of looks like a chess piece. Then it has some white on it, and insects fly around, they get stuck to that sticky trap. Then the one on the right is a different kind of insect trap. It's the red container, the red water bottle, that is full of dry ice. And so the dry ice lets CO2 off. And just like people, animals exhale CO2. So an insect will be flying around and it'll sense that CO2 and it'll fly to it thinking that it's an animal. But it's not an animal, it's my trap. And so this is attracting them to my trap where they get stuck in the trap. Here's one of the moose checking out my traps, seeing what I'm up to. And that CO2 trap there is also powered by a battery. So inside the trap is a fan that blows the insects into the net so I can have them. Then that lunchbox is the battery that's powering the trap. We also set out weather detectors. So those sensors record the temperature as long as they're out. So I can set them to record every 30 seconds. I can record what the temperature is, and I can leave those out for months. Then I can go pick them up, download the data, see what the temperature was where I set my traps. Here's a closer look of the traps. One on the left is that sticky trap. You can see all the insects stuck to it. Just fly around, they get stuck to the trap, and then I have those insects. The one on the right is that trap that used the CO2. So it also attracts them with a little blue light. So that light is up the top there, and they're attracted to that. Below it's a little fan, and it blows them into that net. And then afterwards, I have tubes of insects. So that tube on the left, that was just from me netting a moose for about 2 minutes. I got that many insects. So they're all insects in that tube. Then that sticky piece of paper on the right is all the insects stuck to that paper. And that trap was just out for overnight in Alaska. So I end up with lots of insects. I also use thermal cameras. So that's me holding a thermal camera, looking at the back of a moose. And this looks at different temperatures of the moose. Here's a moose calf. She's trying to help me out, but she's on the wrong side of the camera. And here's what a thermal image looks like. This is an adult moose laying down. You can see all these different colors tell me the different temperature of the moose. So I can tell if she's hot or what her fur does for her. You can also see the insects on the hind end of her, all those little dots are insects. Here's a moose calf, moose baby, similar. Another thing we use is a camera trap. So this is just a camera that automatically takes pictures. So this one I have set to automatically take a picture when an animal walks in front of it. So this moose calf got her picture taken because she walked in front of my camera that was set out there tied to a tree. And that's the mom next to her. Also set it up here in Texas. Here's a white-tailed deer that walked in front of my camera in my yard. Another method that we use is spotlighting. Drive around at night because some animals are out more in the evening. And we're just looking for animals with giant flashlights. Here's a little video of some pigs that we found, some wild boars that we found out while we were spotlighting. So we're just finding animals. Now for bats, so bats navigate and find food with echolocation. So they make these noises and then they listen for the noises and how it comes back to them. And that can tell them where their food is. So we can record these sounds. And these are two different recorders that we use in our lab. The one on the left is a sound meter that's attached to a tree. And we just leave it out all night and it records the audio, the sound of the bat. This other one on the right is a handheld recorder. So we can go out and hold it and listen to the bats by looking at the picture that it gives us. Here's an example of what that might look like. So here's a recording from the handheld bat recorder. One of our students was able to identify this zigzaggy image here, the red, showed that it was a Mexican free-tailed bat. It's a little bit about what it sounds like. So all of these techniques help us learn about the animals and the environment they live in. So where they live, what they're used to, what they're surrounded by, what might be annoying, like these insects, what kind of things they have to deal with, and where animals are. Thank you for listening to my talk.
My name is Annika. I'm a second-year master's student in Dr. Laura Juergens' lab. I'm currently studying the effects of multiple climatic stressors on oyster populations in Galveston Bay. I'm looking at how a prior temperature stress, like if oysters are super stressed out during the summer months, how that will affect them if then oysters are later exposed to a lower salinity stress, like if a hurricane comes through and dumps a lot of fresh water. So now oysters are exposed to one stress, that really hot summer, and a second stress, that really low salinity because of like a hurricane or a lot of rainfall. So what I'm gonna do is I'm gonna show you my setup here, which I set up in the Sea Life facility. So this is the setup for my oyster project. On the shelves here you can see I had 4 different treatments that were at ambient temperature, that were just at room temperature, and half of these tanks were actually at lower salinities. You can see the oysters at the bottom of these tanks. Similarly, you can see that I had 4 different treatments in these water baths, and I set up these water baths so I could keep the tanks at higher temperatures, and the water in these baths were constantly circulating from this larger tank that you will see in a second to maintain that higher temperature.
Hi, my name is Melissa Shashelski. I am the lab manager in the Juergens lab here in the Marine Biology Department at Texas A&M Galveston. I have a degree in marine science and marine biology and a master's degree in environmental science. One of the projects that I work on here in the Juergens lab is the Galveston Bay Biodiversity Monitoring Project. For this project, we put out small settlement panels around the bay, and we will let them grow a small community of invertebrates over a season, so about 3 months, and then we bring them back to the lab and look at what organisms have settled onto that panel and are creating that community across different salinity environments and also time scales in the bay. So one of our sites is here actually on campus in the boat basin, and we're going to pull up one of our panels and take a look at that experimental design. So here's one of our panels that has been out for about 2 months now. So you actually already have a lot of growth happening on here. A few things you can see, these are barnacles over here. And all of these white tubes are serpulid worms. And kind of this pink and slightly orange or brown are a type of bryozoan, an encrusting bryozoan.
And so how do these differ at your different sites?
So some of our lower salinity sites have had fewer species present. In our higher salinity sites, we typically have more species present on the panels. So some of these serpulid worms, we actually have about 5 species that we've found so far across the entire bay. And certain taxa, such as anemones, we only find at our higher salinity sites. So up north, closer to Houston, we haven't found any anemones on our panels there. Some other organisms that we find that aren't on this panel right now, or that we can't see without the microscope, are things like small mussels and oysters, hydroids, which are related to anemones, and other types of worms like spionids and sabellid worms, and we also will find camptozoans, which are a really small colonial organism as well.
Excellent. Have you been able to identify any invasive species on your panels?
We have a few species that we believe are not native. Some of our samples are still awaiting taxonomic verification, but this bryozoan here with the pink, we believe is a non-native species, and actually one of the anemones may be non-native as well.
So that's all really interesting information. Why is it important to study these invertebrates in an estuarine system such as Galveston Bay?
Great question. So these small invertebrate communities are important habitat, but also a food source for a lot of small fish and crabs. So for example, we see mullet and blue crabs both feeding off of not only the panels, but the community that's also growing here on the side of these docks. So they're an important contribution to the ecosystem as a whole, and our monitoring them over time not only establishes what the biodiversity is in the bay, but we can then look for changes in response to either extreme events like hurricanes or the introduction of a non-native species and how that community responds to those stressors.
Hey guys, thank you so much for coming to our virtual booth here at Texas A&M's annual Darwin Day celebration. We are members of a few of the ornithology labs here at Texas A&M, particularly the Grace Lab and the Volker Lab in the Ecology and Conservation Biology department. Now, ornithology, or the study of birds, has been going on for centuries and even influenced Darwin himself as he came up with the basis of evolution as we know it today. Many ornithologists will actually band birds, which is essentially putting a small ring on a bird's leg to track its movements and migration, and so that we can just learn the basics of bird health. So today we wanted to show you all some of our basic field techniques, including banding. And then we also want to showcase some of the amazing bird diversity with specimens here at the Biodiversity Research and Teaching Collections at Texas A&M. We really hope you'll have fun today and check out some of the other awesome booths here at Darwin Day. All right, Gigs, hope you enjoy.
All right, so we have set up a couple of mist nets here behind the BRTC, and we've set up these mist nets right beside a hedge of privet, and the privet berries are favored by the cedar waxwings. And we were lucky enough to just catch a few of these cedar waxwings in this net. And I am going to go ahead and extract this one now. And the first step is to figure out which side of the net the bird came in on, which is pretty simple to do, looking at the underside of the bird. And I am just getting its feet untangled right now and gently pulling the net away from its tail. And now we just have the net around both of the wings, and the net will just be able to loop up over the shoulder of the swing pretty easily here. And I'm going to slide my left hand around and then just pull the rest of the net off of this bird's head, and he or she is now free from the net, and we will take it back to the table for processing and for taking some data from it. Alright, so now that we've extracted our cedar waxwing, we're going to come back to our table or our processing station, and we're going to take some basic body measurements and also look at the bird's health and size as far as what it is within species. So looking at this, a lot of times we can find out a bird's age, at least on songbirds, by looking at the wing feathers, particularly these primary coverts right here, and then sometimes also these tail feathers, which we call retroces. This bird's not wanting to spread them for us, unfortunately, to show that this is, in our case, probably an adult bird. And we're going to be then writing that down on our data sheet and then processing it by banding, taking basic body measurements like tarsus length, which is the length of our leg bone, and then wing cord length, which is the length from the wrist to the tip of the primaries. So let's begin. I already have our band set up here. What I'm going to do is very gently hold this bird's leg steady so I can see exactly where I'm putting the band. This bird is wanting to grab the pliers and poop. Comes with the job. So I'm going to very gently just squeeze the band closed, and I like a few little pulses so that I'm not actually over squeezing the bird's band, and then you have to take it off, and it's a whole process. So a few pulses just till you see it come cleanly together. Alright, so now that we've banded our bird, which is the most important part, we're going to start taking some body measurements. So again, we're going to hold that leg in place. And then, it's kind of hard to see, on the bottom part of this leg, there's a little notch where the tarsus, or the bone that we're measuring, begins. And I'm going to just kind of look at it and see exactly where it goes and measure with my calipers. So, in this case, our bird has a tarsus length of 17.2 millimeters. And I'm going to move on to wing cord. In our lab, we do flattened wing cord, which is a measurement where we're placing the bird's wing completely up against this wing bar, which is essentially a small ruler meant for steadying a wing. And we're just going to press down the primaries enough to keep it flat against the bar. So with this one, it looks like we are at 94 millimeters. Alright, so now that we've banded and measured our bird, going to take a few seconds just to assess its overall health by doing body measurements, so muscle measurements on the breast tissue, and then also checking fat. And we do this, which is a little bit funny, by actually blowing on the bird. To see fat that gathers around the abdominal area and also up on the neck and the collarbone area and underneath the arms or the wing. So typically we go on a scale of 1 to 4, and in this case this bird is going to be about a 1 on fat. And then we're going to also measure the breast muscle, and that tells us pretty much how healthy the bird is, how strong the bird is, and a lot of times this will fluctuate right before migration. So I would call this bird like a 1.5 on muscle score. And that's pretty much how we process a bird. And now we're going to take our time, put our mask back on, take our time and gently release. She is really holding on to my fingers. Just release our bird. So there we go. I hope y'all learned something and have a great time at the rest of Darwin Day.
So hi, I'm Verdiana. I'm the 4th year PhD candidate in the Dr. Grace lab. And we study stress, which is a response to actual or perceived threats to homeostasis. And the hormone that is released in response to stressors is corticosterone in birds. So the way that we take these stress samples or hormone samples is by taking blood. So we take blood samples, so these hormones are circulating in the bloodstream and they are released after probably around 3 minutes after encountering some sort of external stressor. It can be a predation event, it could be being held at some point.
This is where we house the main collection of birds here at the Biodiversity Research Teaching Collections. Our current curator is Dr. Gary Volker in the ECCB department, and the avian collection boasts of over 25,000 specimens. I've pulled a few here to showcase our collections, which not only shows some residents and migrants both in the state of Texas and U.S. Like here, I've pulled the winter migrant, the cedar waxwing. You can see they're really, really beautiful. This was what Michael and the others caught in the nets earlier. Also a spring migrant. This is a painted bunting. Really, really gorgeous. And then we have lots of birds from around the world. So you've probably seen an American robin here, which is very different from the European robin, which you can see is much smaller and is actually in a different family. American robins are in family Turdidae, and European robins are in family Muscicapitae. Then we also have here the white-throated dipper. This is a really amazing songbird. It is the only songbird that is able to hold its breath and dive underwater. Then some really, really amazing tropical birds. We have the lesser bird of paradise, as you can see, incredibly beautiful. Birds of paradise are well known throughout the world for their amazing plumage. We also have the turquoise-browed motmot, which you can see here has beautiful long feathers that lose their barbs, and then those are used for display during the mating season. And then finally we have the double-collared sunbird. Sunbirds are convergent with hummingbirds, which, you know, hummingbirds in North and South America are nectarivores. These guys fill that ecological niche in Africa, and so they've developed the same curved bills and really bright and beautiful plumage. So we use these amazing collections to teach ornithology out here at the BRTC. Thank you for coming along with us and seeing what we have.